This video will explore protein synthesis, a fundamental biological process. There are three main kinds of RNA. Ribosomal RNA, or rRNA, is found in ribosomes and provides the assembly site for polypeptides. Transfer RNA, or tRNA, transports amino acids to the ribosome and positions them correctly. Messenger RNA, or mRNA, is transcribed from DNA and directs the amino acid assembly. The central dogma of molecular biology states that information flows from DNA to an RNA copy, which then directs the assembly of amino acids into a protein. Understanding the genetic code is crucial. How does the order of nucleotides in DNA specify the order of amino acids in a polypeptide? Experiments by Francis Crick and colleagues determined that the genetic code uses codons, which are blocks of information. They hypothesized that a codon consists of 3 nucleotides. This is because 2 nucleotide codons would only yield 16 combinations, which is insufficient for the 20 amino acids. A 3 nucleotide codon allows for 64 different combinations, which is more than enough. Crick's experiments involving nucleotide deletions and additions confirmed that code words have 3 letters. The genetic code is practically universal across all organisms, meaning a codon specifies the same amino acid, whether in bacteria or humans. This universality allows genes to be transferred between organisms, which is utilized in commercial products, such as manufacturing insulin. However, the code is not entirely universal. For example, in mammalian mitochondrial genomes, some codons read differently than in the universal code. For instance, UGA is read as tryptophan instead of a stop codon, and AUA is read as methionine instead of isoleucine. Gene expression involves 2 main steps: transcription, where DNA is copied into RNA, and translation, where RNA directs protein synthesis. Transcription is carried out by RNA polymerase, an enzyme that produces an RNA copy of the DNA sequence. Bacterial RNA polymerase is large and complex, consisting of 5 subunits, each with a specific function, such as binding regulatory proteins or the DNA template. Only one DNA strand, the template strand, is transcribed. RNA polymerase adds ribonucleotides to the growing RNA chain's 3' end. Eukaryotes have 3 different RNA polymerases. I synthesizes rRNA, 2 synthesizes mRNA, and 3 synthesizes tRNA. Transcription starts at RNA polymerase binding sites on the DNA called promoters. These are short, untranscribed sequences that vary widely in efficiency. During initiation in bacteria, the sigma subunit of RNA polymerase recognizes the promoter sequence and binds the enzyme there. In eukaryotes, the process involves multiple protein factors. In elongation, RNA synthesis occurs without a primer. The unwound DNA, RNA polymerase, and growing RNA transcript form a transcription bubble. The transcription bubble moves along the DNA, synthesizing RNA. Unlike DNA polymerase, RNA polymerase lacks proofreading, resulting in more copying errors, though this is generally not harmful. Termination occurs at stop sequences, which cause the RNA polymerase to release the DNA. The simplest stop signal forms a GC hairpin, followed by 4 or more U ribonucleotides. The hairpin causes the polymerase to pause over the uracils. The weak UA base pairing leads to the dissociation of the RNA from the DNA, stopping transcription. Translation begins when mRNA binds to an rRNA molecule in a ribosome. A tRNA molecule with a complementary anticodon binds to the exposed mRNA codon, adding its amino acid to the polypeptide. As mRNA moves, successive codons are exposed. tRNA molecules continuously add amino acids to the growing polypeptide chain. Activating enzymes, specifically aminoacyl tRNA synthetases, are crucial for translation. They pair specific amino acids with the correct tRNA molecules and their corresponding anticodon sequences. Some activating enzymes recognize only one tRNA, while others can recognize multiple tRNAs that code for the same amino acid. 3 codons, UAA, UAG, and UGA, are nonsense codons, serving as stop signals with no corresponding tRNA. The start signal is the codon AUG, which also encodes methionine. Eukaryotic initiation is similar to prokaryotic, but uses methionine as the starting amino acid and involves a more complicated initiation complex with many protein factors.